Hi everyone. We would like to welcome you all to our webinar focusing on MolBoolean, a new brand new method for the analysis of protein interactions in cells and tissue. My name is Mikael Malmqvist, and I'm a principal scientist in the R&D department here at Atlas Antibodies. It is my pleasure to introduce today's presenter, Professor Ola Söderberg, who is the head of the Department of Pharmaceutical Biosciences at Uppsala University. Over the last few years, we've had the pleasure of working closely together with Ola and his research group. Ola has been instrumental for guiding us at Atlas Antibodies in the development of the MolBoolean product that we recently launched to the market. Today, Ola will be talking about the development of the MolBoolean method. And after Ola's presentation, I would like to take the opportunity to highlight some of the key features of the MolBoolean product that is offered by Atlas Antibodies. If you have any questions during the presentation, please type them in the Q&A section, which you will find at the very bottom of the screen. We will gladly answer all questions at the end of the presentation. So without further ado, I will pass the word over to Ola Söderberg. Please.
Thank you very much, Micke. I will just try to share the screen here. So I'm happy to be here and talking to you, but it's a bit sad that I don't see you all. I think that is a downside with the Zoom and not seeing you in real life. So I have been working with method development for many years. So I joined Ulf Landegen's group almost 20 years ago, and then we have been developing many different methods like the in situ proximity ligation assay and many variants thereof. So this is how I spent the last 20 years on my research activity. I started in the beginning as a tumor biologist, so I was working with leukemia, and I saw the needs of better methods to look at protein interactions to understand signal transduction. So when you study cells, you can look at protein expression. So in this case, this is a pool of protein A and this is a pool of protein B. So you can stain with immunofluorescence or immunocytochemistry to see how much of the protein A you have in the cell and how much of the protein B you have in the cell. And that will give you valuable information, but it might not tell you much of how the protein exerts its function in the cell. So proteins usually exert their function as a complex. So the proteins bind to each other. So back then was trying to develop a method that allowed us to look at how big pool of the proteins that are really interacting. And to do this, we developed this in situ proximity ligation assay. And the method, in this case, you can see all the protein interactions will be visualized as single dots. So every little red dots here represent one protein interaction between two molecules. So this gets a single molecule resolution with a standard microscope. And the methods, this method and all the other methods we based on has 3 steps. So the first step is targeting the protein interaction of interest. And for that, we use antibodies. And to these antibodies, we have attached short oligonucleotides. And the different colors here is different domains of these oligonucleotides. So we have the targeting, the antibodies bind to the protein complex, and then you can wash away the excess protein. So, and if it would be one protein, then one antibody would bind to a single protein. But if it's an interaction, you have 2 antibodies binding to the protein complex. The next step is proximity sensing. So we want to see if these 2 antibodies bind close enough to each other. And to do this, the proximal binding will allow the formation of a circular DNA template. So you add 2 oligos, like this from B prime to C prime, I will show later on, and then another oligos that goes from D prime to A prime. So if they bind close, if the antibodies bind in close proximity, the oligos will hybridize to both of these antibodies, and thereby allowing the formation of a DNA circle with ligation. The next step, we utilize this circle as a way to amplify a signal, to get a signal. So once we have created this circle, we can add a DNA polymerase, 529 DNA polymerase, that will start replicating the circle. And you end up with one long single-stranded molecule that consists of repetitive elements complementary to the circle. And this long single-strand DNA molecule is still attached to the antibody that still binds the protein of interest. So everything is done at 30 degrees or 37 degrees. This long rolling circle amplification product will consist of up to 1,000 repeats of the circle. And then if you hybridize a fluorophore-labeled oligonucleotide to this RCA product, you will get 1,000 fluoropores per every molecule. So that means you get a very bright signal. So this oligo design for this, we usually, when we design a method, we usually draw it like a piece of paper, and then we call the different domains A, B, and the complementary, reverse complementary will then be B prime, A prime. And then we can just put in different oligos. Back in the days, we did it manually, trying to design a good oligo system, but now there are also software that can help you with designing the oligos. And over the years, we developed methods that make the rolling circle amplification more compact. If we have variants to solve the problem with dynamic range, so the rolling circle amplification product is around 1 micron in size. So it means that you can only fit a certain amount of signals per cell. So by color coding them, you can fit more. So this is one way to improve the dynamic range. We try to have multiplexed version that you can look at protein A towards many different variants, protein B, C, and D. And also approach is to look at more complex or more than 2 proteins. If you're interested in how we think about this oligonucleotide design and method development, we wrote a review trying to describe the thinking, the thought process behind the method development we do in the group. So the problem now is like you have different methods to look at different things. You have immunofluorescence to look at the amount of protein in a cell, how much protein A you have and how much protein B you have. And in CTPLA will give us information how much protein complex. But of course, it would be good to combine this to have a more deep understanding of what happens in biology. So I usually use a metaphor explaining this, like if you look at the reviews for a restaurant, so you get the 10 people who are saying it was an excellent restaurant. That gives you some information, but of course it doesn't say anything of how many people who answered the survey or what was the other question. So if you really want to understand if this is a restaurant that I should go and try, you wouldn't want to know what was the percentage of people who thought it was a good restaurant. So the idea with MolBoolean was then trying to combine all this information. You get information of both how much free protein you have, protein A and protein B, but also information of how much complex bound protein you have in the cell. And this is, of course, a dissociation constant. So for MolBoolean, we wanted to have information both of the total pool of proteins, protein A, and total pool of protein B, but also how big percentage of these pools are involved in the protein-protein interaction. And this method we published 2 years ago, so we have been working with it for many years. I think it's almost 8 years in the end, and in the last years we worked closely together with the Atlas Antibodies. So we're very grateful for their help in this. So the method is based on oligonucleotides that are attached to antibodies, similar as in CTPLA. But in this case, we hybridize a preformed DNA circle to these oligonucleotides. And the oligonucleotides that are attached to the antibodies consist of a hairpin structure and loop. And the design of the oligos were designed so that they consist the site for a nick case. So we have the oligonucleotide sequence here will be the recognition site for a nick case, that is a restriction enzyme that only cuts one of the strands. So this structure you see here is the loop here. So if the circle hybridizes to the oligonucleotides on the antibodies, then they will become double-stranded. You have a double-stranded stretch, and that is required for the nick case to be able to cut. So the nick case will cut at this position here, that will be at this position here. And also there's the same recognition site also on the other side, so it can cut here. So if you have a protein interaction, the circle will be cut in 2 places. And then you can add 2 complementary oligos that could invade the hairpins and position themselves within the gap formed by the nick case. And this we can then seed with ligation, and then we recreate the circle, but now we equip it with 2 different tag sequences, B' and E'. So if we then do a rolling circle amplification that is primed by the 3' end of the oligonucleotides, either this one or this one, we end up with one long concatenameric product that we can stain with either 1 fluorophore labeled oligo or 2 fluorophore labeled oligos. So I will show you more in detail what happens if you have 1 or 2 proteins bound. So this is exactly the same sequence. Now I draw it more schematic, so I avoid the small hairpin structure that we use just to make the circles. So they have no function in the method itself. So if the circle hybridizes to 2 antibodies binding to protein A and B, you can cut the circle at 2 positions. If the circle hybridizes to only 1 antibody, you can only cut it in one site. And correspondingly, if it hybridizes to the other one, you can cut at this site. So you nick the circle and you add the tags, and the tags will position themselves in the nick. You can ligate together, and in this case you see that you incorporate 2 tags if you have protein interaction and only 1 tag if you have free protein. You do the amplification with 529 DNA polymerase, and you detect with having different detection oligonucleotides staying within 2 different fluorophores. So you can have FITC in green and Texas red in green, in red, for example. So in this case, the rolling circle product will be both red and green. In this situation, only red. In this situation, only green. So this allows us to get to see both protein interactions and free proteins at the same time. So I will give you 2 small examples. So in this case, we culture epithelial cells, and we stain for ecadherin and beta-catenin. So, and then with the detection oligonucleotide that is used for ecadherin will be the magenta, and the beta-catenin in green. So all the rolling circle amplification products that will be formed will then be white. That is a mixture of magenta and green. So this is what we do in the image analysis software. So of course, when you look at the microscope, it's just fluorescence or not. So then you can see that in the junctions between the cells, you have a lot of ecadherin beta-catenin interactions. So that is when the ecadherin from each side meet each other. So if you then induce EMT with the TGF-beta, you will the cells will break up the connections between each other, and you lose the ecadherin beta-catenin interactions in the cells. Another example is emerine and lamin B1 that bind to each other and in the nuclear envelope of the cells. And then we stain then for emerine. We can also see some staining of ER, so that is another position where you should have emerine. And the lamin B1 is also present at low levels in the nucleoplasm. But then you see in the nuclear membrane, you get this very nice staining where you see all the white dots. So if you compare the 2 methods, so both in situ PLA and MolBoolean will of course give information of how much protein interactions you have in a cell. But MolBoolean will also give you information of how much of the two different proteins are expressed in a cell. So you get like seeing the fraction of the protein involved in interaction. In this case, these two images, they are taken by different microscopes. So the in situ PLA is with the epifluorescence and the MolBoolean with the confocal. So there are some differences in sharpness, but usually we think that you get a bit sharper dots with MolBoolean, and I think that is because you have more secondary structures in the circle. So we have seen before that if you can compact the rolling circle amplification product by having oligo that brings distal parts together, but in MolBoolean that is already incorporated in the oligonucleotides design. But they show similar things, but not completely overlapping. So there are some advantages with MolBoolean. And this is the people who has done all the work. So it's I haven't done much lab work myself, and it's mainly Dorothea Raikova and Despina Kempatsu that has done most of the work for the MolBoolean. So with that, I would like to thank you for your attention, and I leave the word to Micke.
Okay. Let's put my camera on as well. There we go. Thank you very much, Ola, for a really great talk highlighting the molecular principles of the MolBoolean technology. I would now like to finish off this webinar by highlighting some of the key features of the MolBoolean product that is offered by Atlas Antibodies. So starting with the question, who is the researcher and who benefits from using MolBoolean? Well, it has been estimated that more than 80% of all proteins do not operate alone. Rather, proteins mediate many of the biological activities in cells by interacting with each other, within cells or between cells. So the protein-protein interactions, PPIs, play key roles in predicting protein function, cell-to-cell interactions, and druggability of molecules. And elucidation of PPI networks also contributes to the analysis of cell signaling events. And we know that the dynamic mechanisms of protein-protein interactions in cells are heavily dependent on the spatial location, but also the abundance of the respective interacting partners. Therefore, it is very important to not only understand the number of interaction events, but also the number of ingoing components. And with MolBoolean, you can study such events in both tissue and cell samples. As Ola has clearly described, MolBoolean differs from other methods in that you can study free and interacting fractions of two endogenous protein targets simultaneously. So by applying the MolBoolean technique, we can monitor the cellular distribution and protein complex formation of important disease markers in cells and tissue, and therefore improve our understanding of diseases. This can provide the basis for translational research to discover new therapeutic approaches within drug discovery, where MolBoolean can be used to elucidate the mechanisms of drug action on a protein-protein interaction. So the MolBoolean kit consists of 15 components, and everything you need to perform the MolBoolean assay is included in the kit, except the primary antibodies. So the primary antibodies are the choice of each customer to add. You can use a standard immunofluorescent microscope, either epifluorescence, or you can use, but it's better to use confocal, obviously, to get more sharper, crisper images. And on the product page, you can find instructions for use and an animation video. There is also an image processing, image analysis tool, and an image analysis tutorial video available. And the method has been validated in tissue and cells. So these examples were already shown by Ola. Again, this is to the left. This is an example where we've investigated interaction between E-catenin and beta-catenin. And you can see these fluorescent objects that Ola mentioned in his talk. We call them rolling circle products, or RCPs. And some of them are fluorescent in only magenta and others in only green. But some of them are fluorescing in both channels, and they will appear white when they overlap in this image. And then you use image analysis to quantify the single versus dual fluorescent RCPs. and the fractions of interacting versus free proteins are presented here as a percentage of total target proteins in the sample. And to the right is the other example that Ola showed, is the analyzing the interaction between emerin and lamin B1. And one can observe protein proximity of the two proteins in the nuclear membrane. But what is also evident is that the proteins are present on their own in the endoplasmic reticulum and the nucleoplasm, respectively. MolBoolean is also validated in FFP tissue. Here to the left, let me get my pointer going, maybe it's better. To the left, we have analyzed the interaction between the ACE2 receptor and the serine protease TMPRSS2 in human kidney. These two proteins were heavily researched during the COVID-19 pandemic because we know that the ACE2 receptor is the protein that the SARS-CoV-2 virus binds to, and TMPRSS2 is the protease that cleaves the viral capsid proteins to enable internalization into human cells. And using MolBoolean, we determined that 32% of all ACE2 receptors and TMPRSS2 proteins are present in a complex with each other. To the right, we have looked at the interaction between SATB2 and HDAC1, and they interact in the nucleus of glandular cells in human colon. You can also perform MolBoolean in distinct cell subpopulations. You can combine MolBoolean with indirect IF. And what you do then is to stain a specific cell type marker protein using a primary antibody from a third species that is different from the mouse and rabbit antibodies that are used for MolBoolean. Or you could use a mouse primary antibody of a different isotype. And in this example, we have performed MolBoolean to analyze the interaction between adenosine A2A receptor and dopamine receptor D2, specifically in astrocytes stained with an anti-GFAP antibody. And during MolBoolean image analysis, you can choose to only quantify objects that overlap with the staining of the astrocyte marker protein, like this image is showing. So we have quantified the number of interacting objects in red here, and the free Adora A2A in blue, and the DRD2 objects in yellow, but only in areas where you are overlapping with the GFAP staining. So this slide highlights some of the main benefits of using MolBoolean to study PPIs in cells and tissue. As we have already mentioned today, MolBoolean offers a more complete spatial quantitative analysis of protein-protein interactions, enabling the detection of both free and interacting proteins. And because of this, MolBoolean offers a more accurate quantification of interaction data by enabling the normalization of the total target protein levels. You're getting biologically relevant data generated with MolBoolean without the need for engineered protein expression. So MolBoolean can then be used to detect endogenous proteins. And because of the rolling circuit amplification that is performed during the MolBoolean assay, you're generating a 1,000-fold increase in fluorescent signal intensity. In addition, the molecular process that is used to generate the amplified products originating from free and interacting proteins, that process is the same for all of the objects generated in the MolBoolean, ensuring uniform signal efficiencies. And the universal kit format of MolBoolean makes it adaptable to different needs with the customer's choice of primary antibodies. And as I've shown you, the MolBoolean is validated in cells and tissue, ensuring reliable data in different sample types. So finally, I would just want to finish off to highlight and show you an example that illustrates the ability of MolBoolean to normalize the interaction data to total target protein levels, something that's difficult to do if you're performing, for example, in situ PLA, in situ proximity ligation assay. We know the importance of using loading controls when performing any form of quantitative immunoassays. For example, we always use a loading control when we compare protein levels between two conditions in Western blot analysis. And this is, of course, also important for in situ protein proximity studies. For example, a drug cell treatment could result in changes in protein expression levels or cell morphology, something that could lead to an increase in total proximity events simply because you have more proteins available per cell to interact. And this is the same image that Ola was showing, but I was just showing you the quantification of the data as well. So in this experiment, showcases the benefit of being able to normalize the interaction data. So TGF-beta was used to treat Hackett cells, and TGF-beta is known to disrupt the interaction between E-cadherin and beta-catenin, and it induces loss of cell-cell contacts at the cell junctions, and that results in a morphological change with an increased cell area and therefore an increase in the total number of RCPs per cell. So if you, like with the in situ PLA, only looked at the amount of interacting proteins per cell, then you may get the wrong impression that TGF-beta actually increases the interaction between E-cadherin and beta-catenin. But in actual fact, the only reason for the increased signal after TGF-beta treatment is the increase in the total number of E-cadherin and beta-catenin proteins that are available per cell. And the ability of MolBoolean to detect all protein fractions, both the free and interacting, allows us to normalize the interaction data and to present it as a fraction of total available target proteins per cell. So we can then see that in actual fact, TGF-beta decreases the fraction of interaction between E-cadherin and beta-catenin, because you have a increase in number of free E-cadherin proteins per cell. So with that, I would like to thank you for listening to us today. We hope that you have learned a lot about how MolBoolean was developed and how it may be used in your research to study protein-protein interactions. I wanted to let you know that we are at your service and that you can write to us at any time. We're happy to answer all your questions regarding MolBoolean or any of our other products for that matter. You could write to support@atlasantibodies.com. And we'll see if we have any questions now in the Q&A section. Let's have a look. So we'll start with the first question. So does this method only help in identifying the interacting partners, or can we quantitate the extent of interaction as well? So I will leave that to you, Ola, to answer.
So I think it's this is also an antibody-based method. So you have to decide, like in advance, what the proteins you're interested to look at. I mean, so two different proteins. So in this case, we showed like E-cadherin, beta-catenin, for example. So it doesn't give you information on other proteins except them, but what it does is it's give you information on both the total amount of the two different proteins and the fraction that are interacting. Then of course it's not like it's an antibody-based method, so it's not a definite value that you get. So it's like you will never detect all the different proteins with antibody-based method. You will see a fraction of protein A, a fraction of protein B, and then you see for these fractions how big part is involved in an interaction.
Okay, thank you, Ola. Moving on to the next question then. Thank you for a wonderful full presentation, Ola. You gave a seminar at Brook University a few years ago, and it is wonderful to see how you have advanced the technology for protein-protein interactions. We commonly employ Atlas antibodies in our work and have successfully employed in studying notch transcriptional condensates. My question is whether you have insights into the efficacy of the MolBoolean approach in the context of phase-separated protein assemblies. We have, for example, observed differences in antibody accessibility. Thanks and all the best, Sasha.
Yes, so I think that is, of course, one of the things one always have to consider. How does the antibody penetrate the cells, and does it bind to the protein of interest? And that will, of course, that can of course differ between different structures within the cell. So it's always easier to target in membrane proteins, and it can be more difficult in the nucleus, for example. But that goes for all antibody-based assays.
Thank you. Next question then. Could you comment on the scenarios where it would be beneficial to use MolBoolean over other proximity assays?
I think it's so there's always, I think Mickey brought this up a bit, like if you have a situation where you have a change in protein abundance, then of course if you're using another proximity method, you will see the decrease in or increase in protein interaction that might just reflect the abundance of the two different proteins. So what you would like to see if you have a post-translation modification of one of the proteins that will affect the binding. So I think it's this type of scenarios, I think MolBoolean will be useful. And also when you have a very mixed population of cells, you have maybe cells that overexpress protein. So if you have like millions of copies of one protein, they will always be close together. So all the different methods to look at protein interactions, they have a distance threshold when they record a proximity event or not. So it's not a physical interaction. So you cannot see with any method if it's a physical interaction, but you will see if the proteins are close by or the antibodies targeted proteins bind in close proximity. So if you have a situation where you have some cells have a low expression and you get the signals, and then you have really, really high protein expression in one or a few cells, you will get maybe the wrong impression of what happens. So then you can use the expression levels as a cue for saying that you might not be able to trust the interaction events recorded in this cell.
Great, thank you. I think we have so many questions, so we're going to try to answer them all. We'll have to move on quickly here then. So how is MolBoolean different from detecting protein-protein interaction using FRET between two different fluorophore tagged antibodies?
I think it's a similar approach, and I think FRET has advantages. And I think the main advantage with MolBoolean is that you get a signal amplification. So for antibody-based FRET, you have two different fluorophores, and you can get a certain amount of fluorescence out. For MolBoolean, we have 1,000 fluorophores per every molecule. So you get a much brighter signal, so it's easier to quantify.
Yes, great. So can MolBoolean be performed on live cells?
And the answer is no. So this base that you need to put in all the different enzymes within the cell, maybe you can do it on the surface of the cell, maybe you can use MolBoolean, but within the cell, I would not do that. So if you want to study processes in living cells, I would rather go for like a fusion proteins and a FRET-based assay. So there's no method that is best in everything. So you have to look at what is the question you want to answer.
Great. So will MolBoolean be able to determine if it's an interaction or mere colocalization?
So I think it's the same there. So it's we will always detect co-localization. So we have a distance threshold. The oligonucleotide part maybe is 10 to 20 nanometer. So it's more or less in the same range as antibody-based FRET. But it's always co-localization. There is no method that look at interactions, not in situ.
Okay, so this is an interesting question. Does one need to know beforehand, before using MolBoolean, that the individual primary antibodies will work for basic PLA, i.e. that the epitopes recognized by the primary antibodies are not masked when the two proteins interact?
So what we do when we select antibodies, we usually buy several antibodies for every protein, and then we try to see how well they bind. Do they bind the protein of interest, yes or no, when you have all the different controls. But it can be good if you want to select antibodies that you use antibodies that can be used for pull down. So if you can pull down a protein complex with antibodies targeting protein A, for example, can you pull down protein B? Then you know that the antibody is not targeting the epitopes used in the interaction event.
It's a really good question, that was. How did you come up with the name MolBoolean? Is there a story behind this name?
Yes, so Boolean is like the Boolean operator that is used like in this is and or not that is used in mathematics. So we thought this is a molecular approach, molecular Boolean approach, that are the proteins interacting or not. So it's just like a word play.
Yep. Can the antibody affinity impact probe hybridization and the downstream quantification of free and interacting proteins? For example, if the two antibodies targeting protein A and B have different binding affinities, can this impact the quantification of the results?
Yes. So I think it's always we have to keep in mind that it's not like absolute numbers of molecules that we see. So it's based on how well the antibodies bind and all the enzymatic steps. But I think antibodies probably where we lose most. So if an antibody can, if you have an added antibody and then the antibody binds maybe 30% of the epitopes and another antibody binds 10% of alveolar epitopes, there will be a bias. So what you need to look at are there difference between situations. So if you add a drug or stimulus, does it change the relative abundance of complexes compared to protein expression?
Okay, good question here as well. I guess confocal microscopy is mandatory to decipher the signal of true interaction from overlap of two individual signals, i.e. false positive due to lack of resolution or signal stacking on said axis.
Yes, so I think it's of course always better to have confocal. So we do a lot of imaging in epifluorescence, and then we use the convolution to try to get the spots brighter. But it's advantageous to look at in 3D also to try to find these different objects if they are dual stained or single stained.
Yeah,
So it also depends on what you have. If you don't have a confocal or advanced microscope, you will still be able to do it with a standard epifluorescence microscope.
Yes, and we have people have used MolBoolean and they've used a standard epifluorescent microscope. What I wanted to add is that not only do we do the image deconvolution, we also have a module within the image analysis pipeline where you are sharpening the objects before segmenting them. So there are ways of sharpening objects, so you can use also non-confocal images. Okay, moving down the list of questions here. The MolBoolean images are characterized by single or dual fluorescently colored objects, which requires more advanced image analysis. Could you comment something on the image analysis that is carried out to separate single from dual fluorescent objects? And are there any alternative approaches, you think, in the pipeline?
Yeah. So what we do now is that we do like a similar, we do a scatter plot of all the, we defined all the different objects in the image analysis program, and then we plot them as a scatter plot, like for similar as you do for flow cytometry, and try to identify objects that are single color and dual color, and then we put them back, the information back on the image. So it's a bit complicated, but we are also trying to develop approaches where you get them in 3 different colors. So you get free protein A and B in separate colors, and you get the protein interaction then in a 3rd color. So it might be easier in the future that you don't need to have as advanced image analysis as you would do for MolBoolean. So this is something that we're working on, and maybe that's something that that Atlas will maybe have in the future versions.
Thank you, Ola. Let's have a look here. Stephanie's writing, hello, thanks for a nice talk. How do you link the oligos to the antibodies? Do you know the number of oligos per antibody, and does this matter? And do you see a difference between using primary or secondary antibody probes?
So I think it's the conjugation we use like a bifunctional linkers. So you have NHS ester targeting the antibodies, and then you use either you can use this, what's it called, hydrazine or click chemistry to targeting the oligonucleotides. So nowadays we have usually ACID modified oligos, and then we have a DBCO modified DBCO NHS ester bifunctional linker to target the antibodies. So the number of oligos per antibodies we can vary. When we do it, we always purify the antibodies with the HPLC, so we see that the most are only having one oligo per antibodies, but there are also fractions that has more. For MolBoolean, it doesn't matter so much because you have a preformed circle hybridized to the oligo that you then cut and then religate. So it means that you will always come back to the circular molecule in the end. For PLA, it's more of a problem because if you have many oligos, it will also be situations where you create linear ligations, linear ligation protein, even though the proteins, the antibodies bind in close proximity. So for MolBoolean, I don't think it's a problem, but other methods, maybe a little bit.
Okay.
And
Great.
it was something else also, secondary, primary. And it's always best, of course, it's always better with the primary because it's more tight the distance requirement. But it's a cost and ease of use thing also. So secondaries, you can produce a big batch and then you can just swap the primary antibodies.
Yes. So since another question here, so since the customers uses their own primary antibodies, do the oligoconjugated probes function as secondary antibodies? And yes, the answer is yes to that one, right? It's the anti-mouse and anti-rabbit secondary antibodies, yes. Quick answer to that. What is the distance between the proteins that create the reaction? Well, I guess the question would be, what is the maximum distance between the proteins to enable overlapping signal in MolBoolean?
So I think it's similar to the PLA. So it's antibodies with oligos. So for PLA, we estimate it to be around 40 nanometer, the absolute maximum distance if you have the distance between the epitopes where the primary antibodies bind to, and then you have the secondary, and then the oligo. So if you would have primary conjugates, they will be, of course, shorter, maybe we're down to 25. The distance between the oligonucleotides, they are probably around 15 to, maybe 15 nanometers, something like that. So it's a little bit higher than, or like larger than the FRET distance, that is 10 nanometer, but it's within the same range.
Okay, I think this is a very interesting question. Is there any data available about how often a protein-protein interaction may obscure an antibody epitope? I'm wondering if protein visibility may be reduced if the epitope is obscured during the protein-protein interaction.
Absolutely, and I think we have seen that when we haven't investigated thoroughly, but we have used antibodies that doesn't work, so to monitoring protein interactions. There's also situations when you have conformational change when you activate a protein. So I think we did a long time ago, we looked at the EGF receptor, and when EGF receptor binds EGF, you have a conformational change, and it was some antibodies that didn't bind any longer when you stimulate it. So it's always, are the epitopes available? I think it's always a crucial question, and I think the only way to address it is to try. Does it work or not?
And I also want to add there that there are websites out there where you can look at, there's a protein databases where you can look at the tertiary structure of protein-protein interactions. And so you can see where two proteins are interacting in a complex. And it's always good if the primary antibody provider is giving you the actual epitope that your antibodies are binding to, because then you can have a look at this database where the epitope sequence is far away from where the two proteins are interacting or not in a tertiary structure. So again, very good if you know the epitope where your antibodies are binding, but not all antibody providers can provide that, unfortunately. So that's important to know for sure. All right. how does one distinguish the double-labeled interaction partners from non-interacting proteins in close proximity to each other?
And I think we cannot. So
Yeah,
so the method only detects proximity.
Yeah, so we already covered that really.
And post-translation modifications, I saw it there. And that is of course also perfectly fine to study. So if you have an antibody targeting post-translation modification like a phosphorylated tyrosine, and then you can have one antibody targeting the receptor, and you can detect how much of the receptors are phosphorylated or not. So that works perfectly fine as well.
Yeah. And I think you already touched upon this. Does antibody binding affinity affect the method? And you answered already, right? It does affect the method. So you need to have primary antibodies with a good binding affinity. So thank you for a wonderful presentation. For human samples, you mentioned that the assay has been used for FFPE tissue, but has the assay also been tested in frozen samples, and has the assay been used on post-mortem samples or just surgical resections?
So I think what we have tested is FFPE. So, but based upon experience with PLA, for example, we know that fresh frozen tissue sections usually work better than FFPE because you have always the problem with fixation that you lose the availability of epitopes. So I would assume that it would work better on fresh frozen tissue samples. And the post-mortem or surgically resected, I think it's always different what proteins you're interested in. Does it change are the proteins degraded? If you look at the post-translation modifications like that are short-lived, you need to have the tissue as quickly as possible to fixation.
Okay. There's a question here from can we use this for looking at RNA protein interactions? For example, if the RNA fish probe is labeled with digoxygenin and the antibodies used are against anti-dig, which is the RNA then, and perhaps an RNA binding protein. In other words, is the protocol compatible with RNA fish? Thank you.
We have never tried, but I think I would assume that it would be. I mean, if the fish probes are specific and bind well, so you have to adjust the protocol so that it works for the fish probes, and after that, I think it would be perfectly fine to use the MolBoolean as well. I think, of course, you have less RNA in the cell, of course, than you have protein. So it might be that numbers of CNS might be lower.
Yeah.
But I think it's just to try.
And then it's important to have a negative control then to know what is the background signal. So you can subtract that in your analysis. Any advice on choosing primary antibodies? And I think I can answer that. I think it's important, like I mentioned, to if you can find out the epitope where the antibodies are binding, and if you can get any idea of whether those epitopes are masked for the protein interaction. If you don't know the epitopes, you can do a co-IP, like Ola was mentioning, to see whether you can pull down the proteins together and see whether the epitopes are masked when you have proteins in a complex that way first, before you proceed to do the MolBoolean. Yes, it is important to have good primary antibodies where you know where they bind, definitely on the proteins. Can proteins come to, next question, can proteins come to similar proximity to be detected by the assay but not actually interact? Yeah, I think we've answered that.
Yeah, that can be.
Measuring, yeah. You're measuring of proximity, not actual physical contact. Next question. Are antibody oligo complexes available through Atlas? And if yes, what is the cost, or do we need to generate them ourselves? Yeah, so the MolBoolean product contains all of the components you need to perform MolBoolean, other than the primary antibodies. So the antibody probes, which are the secondary antibodies with oligonucleotides attached to them, they are as part of the kit. Yes. So you don't have to generate them yourself. Can you detect multiple interactions in the same cell?
So I think in theory, so I think the size that we use for detection oligo, this is the hairpin structure that is below the in the oligonucleotides attached to the antibody. So of course you can have different variants. You can have one hairpin with a different sequence. So it would be possible to have like protein A, B interacting with C, D, and then you see all the different combinations. But that is not something we have tried yet, but I think in theory, I think it should be working fine. We have done similar things with PLA, so I don't see why it shouldn't, but it will require some oligonucleotide design.
Yes, great. Next question. Can I only use mouse and rabbit primary antibodies? And I can answer that one. Yes, the product that we're offering here at Atlas Antibodies is only anti-mouse and anti-rabbit currently. In theory, you could use any species, so you could have your antibody probes in other species, but that's not offered by us at the moment. Last question, and I think that's the question is I think you mentioned that this approach can be used to look at multiple protein interactions, say between 3 proteins. Is that correct?
So I think it's a different type. So that is we use to PLA, that you can use different proteins, different designs of the PLA assays to look at 2 or 3 proteins. So with MolBoolean, we haven't tried that yet. So that is also something that is possible to do in theory. You just have to redesign the oligos that makes the circle, but that is nothing that we have been working with. I think it's always a bit problem. So we talked about that before, like how big proportion of the protein pools do you bind with an antibody? So if you have, say that you bind 30% with each antibody. So if you look at the interaction, then between 2, you have 30% times 30%, then you're down to 10%. And then if you have 3 proteins, it's 30 times 30 times 30, then you're down to 3%. So it's of course more difficult to target larger protein complexes, and also that the oligos have to reach everyone. So it's more complicated. It's not an easy task.
Okay, that was all questions. Thank you very much for really, really good questions. And thank you, Ola, for answering them all in a detailed way. Again, Ola Söderberg, thank you very much for taking your time to give us this great talk about how you developed MolBoolean. And thank you all for listening to us today.
Okay.
Bye-bye.
Thank you very much.